much read. So, we're still talking about photosynthesis clearly. And so one of the things is next week we're gonna put the book away. Actually, starting Friday, there is a short part from the book, and then we're gonna start reading some more health, climate change. People learn enough about how it's affecting plants. So, like, what does it really mean in terms of the world? And of course, that could be like an entire semester. But we're going to start exploring more how it will affect insects and other diseases and the impact on food security. So we have after today we have 3 more lectures in 4 weeks and 3. And so next week we're going to focus more on articles. And then on Wednesday, because I know just before the midterm I need you're oversaturated with all of these, getting some more like live reads from the New York Times to talk a little bit about food security as it is right now. With the time of the war on fossil fuels and the floods and all of that. So hopefully Wednesday before the exam, for those of you who will have to take the exam, and easier, lighter. Also, that means that we have 1, 2, 3, 4 more lectures before the exam. So 4 more lectures to earn more points. Okay, so a couple of things. The preliminary topic of your final presentation is due today at 6 p.m. The link is posted on Canvas. Please make sure you submit it by today so that I can start giving you feedback. There's a reading assignment, as I was saying, a short portion of chapter 9, and an article that is already uploaded on Canvas for Friday. Also, we're still a month away almost from this, but I wanted to start telling you what this is. So if you remember anything, probably you want from syllabus, is that 10% of your grade is going to be derived from an activity called your mind on plants. And so we'll be talking more about it later on. But basically what it means is that during November 4, 7, and 9, instead of like a traditional lecture, you're going to be doing some research on 3 specific metabolites that alter the human brain, the human mind, and we're going to be having in-class discussions. So there is a description, and I'm just doing some sort of part here. So you're going to be doing literature research. In the first one, you turn in caffeine, which should be a sort of easy find of literature about caffeine. Again, I'll tell you more about it later on. November 7, we're going to focus on opioids also, from plants. And in November 9, I'm leaving it up to you. Is there any secondary metabolite from plants that alters the human mind that you're interested in for some reason? Besides caffeine and besides opioids. Okay. So if you have anything in mind, good. If you have nothing in mind, you can start thinking, so there's simple things that you will think of, like tobacco, like cannabis, and then from there, whatever you like. It has to be to the human mind, so like aspirin wouldn't do it, because we're kind of exploring the effect of secondary metabolites in the human mind. So to give you a brief overview, the idea is that we discuss, of course, the role of that metabolite in plant physiology. After all, this is a plant class. The metabolic pathway involved, the environmental factors that stimulated or inhibited, and any historical issues. So we're going to talk about, just let's say, caffeine. Like, what is the history? Like, eventually someone had to step on some kind of coffee plant related and figure, hey, this keeps me awake. So can you dig out some of the story about that? It's not a history class, which is more like let's come here and share a story of how coffee was discovered, how someone stumbled on that, and how it spread through the world. Also, the history of opioids is very interesting. You'll be surprised that babies were given heroin at some point, and that was recommended, and it was a good thing. So, it's more like, let's talk about this. Yes, heroin was actually created by Bayer at some point, and it was prescribed to people. Uh, so some of these just, more, yes, related to plants, but also kind of make plants look a little bit more interesting, right? And then, so you will see I posted some guidelines because will read, you will be submitting a writing portion on it. That is, for example, due November 4 at the beginning of class, you should have submitted that on Canvas. But you're going to bring that, with you so that you have some material to discuss in class. So there's going to be a written portion related to this. And your grade for this 10% is going to be from the written portion and the class discussion that I'm assuming, because everybody has submitted your assignments, you have material to talk about. Okay? And it's going to be, I'm going to structure it somehow, but the idea is that it's like a student-driven discussion class where we just share what we've learned. Okay? There is a module on Canvas now that I call Your Mining Plans, and there are the guidelines. So start seeing what you have to talk about. And on Wednesday, November 2nd, again, we're still away from that, I'll give you an introduction to secondary metabolism, and I will explain more exactly like an introduction and explain the format more specifically, but if you want to start thinking, because clearly it's going to be 3 papers due back to back to back to back. So you can start reading about it. From now, it's going to be good. Okay? Okay, so let's start with class. So we have been talking about Rubisco. Can you tell me 2 specific environmental factors that affect the carbon fixation mechanism of Rubisco? And how basically this impacts the plant?
Like relative concentration of CO2 compared to oxygen. If you have more CO2, then Rubisco is going to bind CO2 more often. So oxygen makes it more for respiration, which is bad generally. And then sunlight also directly activates Rubisco.
Yes. So, we talked about how Rubisco can do the right thing and the wrong thing. And the right thing and the wrong thing can be altered by different mechanisms. So light needs to activate the activity of Rubisco. We also talked about how higher temperatures basically make Rubisco do more than one thing. So it activates the oxygenation mechanism more than the carboxylation mechanism. How at higher temperatures, CO2 is the air will contain less CO2, is less dissolved in air, so it's less likely that the plant will encounter CO2 instead of oxygen. So in general, under higher temperatures, you're going to have more likely Rubisco doing the wrong. You need to tell me 2 things.
Well, part of it is that, so you have your mesophyll cells and bundled sheet cells, so they're going to do all of their reactions for the Calvin cycle in their bundled sheet cells, where they're going to be there's essentially not going to be any oxygen there. And they're also going to structure the area around when they pull in so that all of the bundled sheet cells are further in and they're separated by those mesophyll cells so that oxygen can't readily get to them for those reactions. And then, so like I guess another difference would be to keep all of their Rubisco for the most part in the bundle sheet opposed to other plants where they keep them mesophyll.
Yeah, so basically there's this separation of space in which you have different types of cells doing different things. And so because you have the slides from before, so I thought this was very kind of straightforward. So in the C3 photosynthesis, you have single type of mesophyll cells doing the same everything. CO2 is fixed by Rubisco and forms this 3-carbon compound, and then just proceeds the Calvin cycle. In C4 pathway, you have C4 because the first stable component is not a 3-carbon molecule but a 4-carbon molecule. We have Rubisco, as Justin said, mostly on the bundle sheet cells in here. And the initial CO2 is not added by Rubisco to something. It's a different enzyme that we're going to be talking about it today. So that 1 carbon is fixed with this 3 carbon into 4 carbon, and then this constant carbon concentration mechanism basically moves into a different cell and hands Rubisco, CO2 in the hands of Rubisco where there's less oxygen and less likely that Rubisco will do the wrong thing because there's less oxygen present here. So one of the most important things is that the first enzyme of the pathway, so the one that's dealing with CO2, the atmospheric CO2, is not Rubisco. You're gonna see it as is hep carboxylase, so just something else that is not Rubisco. And what it actually does is that, CO2 gets into the cell and it is, converted into something else to make it more difficult for you, and then eventually a 4-carbon product. Okay, so then that 4-carbon product, as you can see here, diffuses into the bundle sheet cells, and in these bundle sheet cells, it's handed on to Rubisco. So again, Rubisco does less of the wrong thing, and because it does less of the wrong thing, then it has to spend less time doing the C2 oxidative photosynthetic pathway, which was the photorespiration. That remember, the C2 compound went through 3 different organelles to recover some of the carbon because C2 is also toxic for the plant. So by having this separation, the plant is minimizing Rubisco doing the wrong thing. And so this, I don't expect you to remember this, but I shared this picture in lecture 7, which seems like years ago. And we were talking then about transpiration ratio. And transpiration ratio is how much water is lost, or how many molecules of water a plant loses to fix 1 carbon from CO2 into sugars. That's what we talked about in lecture 7. And so C3 plants, we said that the transpiration ratio was about 400, which means plants will have to uptake about 400 molecules of water to fix 1 C sugar. C4 plants, now hopefully it makes sense more why the transpiration ratio is only 150. It is going to use less water because, water, stomach, more transpiration, all of that is improved by having this kind of separation, which decreases the transpiration ratio and makes plants, many of them in the tropics, much more likely to adapt to warmer. Yes.
A.
Why?
Because they're cacti and they are in arid environments, and they need to photosynthesize during the day and fixate carbon at night.
Yes, so it would be A, right? So that leads us to the last photosynthesis, that is CAM photosynthesis. And this is what CAM stands for, and I'm going to say this again, because I cannot. But basically what happens is this type of photosynthesis was discovered in a succulent plant from this family, and that's why they just call it CAM. Oh, I didn't do your homework.
You want to read it, but you only need to fill out. Okay.
We just have to fill it. One of these rows. Reading.
Initial fixation is Google this out. So atmospheres to C4 is
Initially fixed externality and then
facing by type.
that's just identity is so malady would that be?
Or it's just CO2. Is it pep oxidase?
So yeah, that brings it in initially.
That'd be just like
Versus
cancellation instead of everything.
I saw it also.
So it's good, so we can say like. Yes, so the idea is that eventually Rubisco is important for all of them, right? I mean, Rubisco will eventually put it into the sugars. It's the only one that has the ability to take the carbonated sugars, right? So I'm talking, that's why I said in this case, atmospheric. So
For that one, would it be the enzyme?
yes, yes.
Just wanted to make sure I wasn't ready
Yeah.
to go from all of them.
Okay, so it's just
Yes,
C4,
yes.
but
Separate that time.
That would go for
So we're gonna stay fixing it.
yes.
I think that's right.
Yes.
Well, it's not like there's like
This would be
But like the pathway that
such a big one.
yeah, it's I don't remember the last time we talked about it. Is that we have to go back.
the water.
Oh, what was the conversation? No, there's no chance on the last that. This is the big shape points. So I think you have. Check the screen strip. It might be moving race, but there's a panel.
I can visualize kind of, but it is, I can't think of what
Yeah.
to say.
I know, like there's some pathway to factor.
Yeah, so is it like that?
Well, I think we're going a little bit ahead of this. We're supposed to do this point. Just supposed to go straight on. I think we're going to discuss it a little bit more.
Good call.
Yeah. It also would either be like a butterscotch.
Thank you. Did you do any quarterly or anything else?
Not too much. I went and helped people with some riding lessons for my horses, and that's about it.
So, is it fun?
It's pretty fun, and it's also a good way to help kids who have anxiety or just never been around a large animal.
Yeah. That's super fun. It's really fun.
Yeah.
How old like kids you're
I work with people from as young as 5 to 95 if they want to.
Oh, really?
Yeah.
Nice.
I've had some people who are like in their 50s to come for their first ever riding.
That's cool.
Yeah.
Yeah, that'd be pretty cool. Like doing lessons of any kind is a cool environment.
Yeah.
I enjoy teaching people things.
Yeah. Well, first lessons are always just mostly about the safety of it.
Yeah.
Yeah. I just walk with them most of the time just
Yeah.
in case they get scared or drop a rain.
Don't have to ride the
Yeah.
ride the horse around the ring. I would go from anti-anxiety experience to high anxiety experience very quickly.
Yep. And hey, some people are just terrified of horses for some reason.
They're,
They're gentle giants.
yeah.
Also, I've been stepped on. It's like
Yeah.
when I was like 5, I had my hand stepped on by a horse while I was pulling gravel. Yeah, it was not fun.
Yeah, that's.
I just got a full bucket of water and was told to walk it off. Yep. Well, Nurse Mom was telling me to just suck it up at that point.
Yeah, the farm.
Yeah.
Farm way. That's insane.
But yeah, and even though all that, I never blamed the horse.
Yeah.
He was just chilling, his foot, and
Yeah.
just unfortunate.
Shit happens moment.
Yeah. Yeah. Heck, even some of them, when they're trained, if something sudden happens, they're an animal, they're gonna react. You just train them to react not as terrifying as they could. One time at my county fair, I had a little girl who was leasing my horse, and some kids were playing baseball, and they hit the baseball into the metal bleachers. And luckily my horse is decently trained. She just sidestepped, but the little girl wasn't prepared, and she slipped off. And then started screaming, yelling, crying. And my horse just looked at her like, "Are you okay?" That could have been a double whammy of, "Oh, we're scared now. Time to run."
I know that they have that conference where you can't stand snakes. Is that true or is that fake?
It depends. Some of them, some are afraid of plastic bags.
But
Yeah. Like legitimately, some trainers just make them walk across plastic bags or tarp just to make them be able to handle that.
Huh.
My horse is trained enough she can walk through a door, like a person door.
Right.
Yes, and she has.
That's super funny. Did you like set up a door in a corral, or did you just,
We
you just walk through the door?
We just trained her to walk through a gate, and then eventually we just took her through a door. She was like, "No, this is just a very thin gate."
Yeah, that's great. Horses are super cool. I haven't been around horses a ton. I don't have like a very healthy respect to them. They're powerful creatures.
They're 1,000 pounds muscle.
Yeah, they are. Oh, yeah, you're gonna see a horse like it's like that.
Oh, yeah, those Clydesdale breed is pretty.
Yeah.
They warm blood boys. Those are actually gentle giants. But they could. Yeah.
Just like
They're just big.
powerful, yeah. I think it's like puts humans into perspective.
Yeah.
Like we like to think that we're powerful, but
Crazy that someone was able to tame one of those things just to ride it. Also, fun fact: tomatoes are poisonous to horses.
really,
Yep, they're the nightshade family. Yeah.
That's fascinating. You always know if anything like poisonous to us and like not poisonous to like other animals.
That and shoot, there's a certain type of tree that the leaves fall and like they get a chance to like they don't get a chance to dry up. Those are poisonous. I forget which one. Yeah.
Does it like different creatures metabolize things differently?
Yeah, and the horses are unable to throw up.
Really?
Yeah, but.
So if something bothers them too bad, their stomach will kind of turn and they colic. So it's basically their stomach's just flipped and twisted so much that everything's just bad. That kills them sometimes.
It's like weird that they would like not have a mechanism for that. Like evolutionary.
They never really had to.
Okay.
They were originally
So some of
transported
you
from
are
Spain.
working on question 2, you don't have to do that just yet.
Oh, wait.
So, fine, but let's move on. Okay, so I'm assuming everybody has the answers to at least the next 3 questions or so, or probably 10. But I need to think about it. Yes.
So CAM is similar to C4 in that it's like separated into like a bundle of sheath cells. But it's different because it separates it by like time as well as like that spatial distance. It's also different from C3 in that way because like it's separated by space. And I'd say it's more similar to C4 because it is like the same pathways. Like it's fixing it in the mesophyll and then like putting it through the Calvin cycle and then that bundle sheath is just like separated temporally.
So you think it's more similar to C4. So how is it, well, I think you kind of said, so can you tell me how it is different from C3 just to sum up?
Yeah. Different from C3 because it separates the Calvin cycle into the bundle sheath cells
Okay,
and like fixes carbon in the mesophyll.
okay. So a couple of things there. It is, I would also agree that it's more similar to C4. You can, argue both ways. So there's really a correct answer. My response to why it's similar to C4 is because the first stable product is a for carbon, so I would call it more similar to C4. However, there's something that is C4 separates it in space, CAM in time, but not in space. Okay. So this is happening all in the same mesophyll cell. So it doesn't have a distinction between bundle sheets and mesophyll. So that's why you can argue it could be more similar to C3 because it only happens in one type of cell. That it could be more similar to C4 if you think that there is an additional enzyme and the first stable product is C4. So if it's not separated in space, how is it that this separation still works as a carbon concentration mechanism? If it's the same cell, why isn't Rubisco messing up? Why is it messing up less? Yes.
Because it's storing it as starch, so you just can't use it.
Starch is a carbohydrate. And Rubisco, so this is one thing. Let me clarify. Rubisco is the only enzyme that can make that carbon into carbohydrate. Okay? So eventually it stores it in the chloroplast. After all of this, it will store it as starch. Okay? So that will happen after. It is going to be stored as starch at night in the chloroplast. But let's back a little bit before that happens. If it's the same cell, why is Rubisco not messing up so much? If kind of CO2 is right out the door.
Is it because it's storing it as an acid instead? So it keeps it as like a C4 acid and so that way it can like, I'm trying to word this right. Convert it into the C4 carbon later. So it stores as a 4 carbon acid and then converts it later.
Later, and that's the key. Later when? Why is the Rubisco competing with the CO2 in the atmosphere?
Because Rubisco is like activated and it's doing that at night, so then during the day Rubisco starts to fix the carbon.
Yeah, so the thing is, the key is that something happens during the day and something else happens at night. And so while stomach are open, you can see here at night, right? Stomach open at night. So CO2 is coming in at night, where clearly temperature is not as high and all of that. And that CO2 is fixed by the same type of enzyme that fixes the C4 in C4 photosynthesis. And it is stored as a C4 compound in the vacuole overnight. So let's produce this, open stomach at night, and we have this enzyme active at night. However, Rubisco is not active at night. Okay. Remember how we said Rubisco is still activated by light and all of those things that happen? Well, it's not the same mechanism, but still, during the day, what you have is now that malic acid, so that was stored in the vacuole, is similar to that carbon concentration mechanism that we had in the bundle sheet. So instead of being a separate cell, Rubisco is not worrying about anything. So once stomach are closed, Rubisco is not competing with oxygen in the atmosphere. It's not trying to grab the CO2 from the atmosphere. It's just grabbing the CO2 that is being released by something here, again, in a carbon concentration mechanism. So this head case is active at night when stomates are open. And unlike most plants, stomates are more open at night and then fully closed during the day. Or not fully closed, but somewhat closed during the day. So what can you tell me from this data? I'll let you read that. Yes.
So the assimilation rate for C3 plants when under constant illumination obviously is very low when it's not having constant illumination at the -12 hours per 12 point. But at 72, it stays consistently high throughout the time period. And then for the cam plants, it's kind of like it never will get above 20 typically, and never below 10. So it stays in that consistent range throughout, regardless of the time under illumination.
Okay, yes. So C3 plants, you're correct. It's telling us in dark, there's no carbon assimilation. There's no CO2 assimilation in C3 plants. As long as you have light, apparently carbon assimilation, CO2 assimilation is high. No. And that brings us back to whenever we talked about Rubisco and all of the Calvin cycles requiring light and ferredoxin and all of those reduced ferredoxin to activate. So Rubisco, as long as there's light, is doing what it's just bringing in CO2. Now, you mentioned, how is that different from what's happening here in Camden?
It just kind of fluctuates through a very standard range. Like it doesn't, so in C3, it gets the assimilation of CO2 gets very high over the consistent time period of more light on it. Whereas the cam plants, it stays just in that neutral range of where it is right now.
So it stays in a neutral range, but is it constant?
Not as constant as the C3 plants.
Okay, so what can you, so that's correct. What can you get from that? What does it mean?
Sometimes cycles of lower and higher stimulation, even though there's full load.
Yeah, so it's going on cycles of, as you can see here, regardless of whether there's light or not, it is still behaving in cycles. Can you tell me what that could mean? How is carbon assimilation regulated in CAM plants if it's in cycles of about 12 hours? Do you know how you call that regulation that happens every 12 hours, sort of? Anyone wants to give it a try? That's probably a new term, so it kind of regulates us. So have you ever heard of, so technically if we're, even if we don't see light, there are many things in us. Even if we're exposed to constant light, there's going to be a time when your body is going to tell you, go to sleep, wake up. And there are certain hormones and certain things in us that are going to be regulated because of an internal clock that many things are regulated by. Yes.
I know it's like humans call this circadian rhythm.
Yes, so circadian rhythm, okay? So in C3 plants, carbon assimilation is regulated by light. In CAM plants, it's regulated by circadian rhythm. So basically, light or no light, CO2 is going to be used. And so remember, one thing is HEP and one thing is Rubisco. But basically, it's going to be regulated by this internal clock that is telling the plants pretty much what to do and when to do it, regardless of what's happening outside. So that's, and so up until now we've been talking about all of these photosynthesis, but now we're going to step outside of the cell a little bit, kind of see how environmental factors can affect photosynthesis, and in turn more than photosynthesis, the physiology of plants. Now we know what photosynthesis means for plants, so what environmental factors. I'm going to show you a series of slides, and you're going to tell me what you see. What do you see? What did you see?
Plants closer to the light grew taller and had more flowering than the ones farther away.
Yes, so what you're seeing here is plants that are closer to the light. It's not very clear here, but they grow different. And not just the height, but also the developmental stage. They are flowering even faster than these other plants. That is telling you that same plants, same environment, like temperature and all of that, still there are other factors that can regulate how much they grow. In this case, it would be light. Yes. Yes. One. Can you tell me something more about it? Like, why one?
It's because it's a limiting factor. So only one thing can limit it because it's going to be at the bottom limiting that one factor.
Yeah. So basically, what happens is that this is known as the blackness. Okay. And so it is telling you that there's a limiting factor. So what is the most limiting factor here? So here in this example, what you have is, the rate of photosynthesis and CO2 concentration and just different levels of light intensity. What you can see here is that there are certain light intensity, right? When you have this, let's say, E level of light intensity. So even if you increase CO2 concentration, the rate of photosynthesis increases a little bit, but at some point, if you don't increase the light, even if you increase more CO2, the rate of photosynthesis is not. So in this case, light is the limiting factor. You increase light a little bit, which we know is important for CO2 fixation, and then you increase light, and so the rate of photosynthesis increases, but eventually, even when you increase the concentration of CO2, the rate of photosynthesis doesn't increase anymore, because basically the limiting factor in this case is going to be light. You have light, you have the enzymes ready to go, but there's no more CO2 basically there or a higher rate for the plant to increase the rate of photosynthesis. Why do you think knowing this is important when you're studying plants? Any kind of plant experiment? Yes.
Well, it can be useful for keeping them alive, just like in general.
Yes, and if you're experimenting with plants, why would it be important? Yes.
I mean, like, say you want to identify the growth rate of a plant, you can maximize all of its, maximum sunlight, give it enough CO2, but eventually at a point, it's only going to go as fast as it can produce those carbohydrates. So you'll learn about like how well it can actually, or how much those enzymes are available to do that set of reactions. So that's important.
So basically, when you're measuring anything, any kind of experiment, you have to be aware of what I can only be limiting one factor. So if I'm trying to see, how much faster this plant grows with fertilizer, without fertilizer, maybe I give it so much fertilizer that the plant can now thrive, and maybe I'm not really measuring how much it can grow because now it's growing so well and light becomes the limiting factor. So basically you can only change when you're doing any kind of plant study. You will have to either limit light or limit CO2 or limit water, but you have to make sure all of the other conditions are maximizing whatever. They are not the limiting factor, and the only limiting factor is what you're giving. So for example, if you're measuring light periods and CO2 and water, you might think that if you do a very complex mathematical model, you can try to figure out how all of these are affecting and converging into something, but the reality is only one factor, whatever limiting it is, and you won't know because you're measuring three different ones, is the one that is limiting your plant growth. So whenever a plant is undergoing or we're measuring photosynthetic performance, there are three major processes that can be limited or that are going to regulate the photosynthetic performance. And it is the Rubisco capacity. So how fast Rubisco can do. How much that 5-carbon molecule can be regenerated. Because yes, you want Rubisco to do the right thing, but remember that 5-carbon molecule that is regenerated to attach to that 1 carbon from CO2. That also needs to be regenerated. And how fast can you produce those sugars to basically give you starch and give you sucrose for the plant to grow. So it is all demand and supply of photosynthesis. So demand by how much light is present, how much CO2 is there. But also remember at the end how much CO2 can come in and do all of this work. It's also regulated by the guard cells that are going to be opening or closing stomach to allow more or less CO2 to come. So this is influenced by leaf properties. So if you have too little of something, let's say too little light, or too much light, or too cold, or too hot, or too something, or too little of something, we know that like animals, plants can adapt to some extent. The difference is animals, for the most part, can go and move, and it's too hot, let's go to the shade, let's go have water. Plants cannot afford to do this. So they have different strategies to adapt.
So it's really just-
Low light.
I'm pretty sure like sunny so then adjust like direction. And so over the super sunny,
No, so every time, so you mean when it comes out of the cutting cycle?
Well, I think the products to see.
Oh, okay, this product, yes, so it would be, yes, yes, I thought you were talking like product at the end, yes.
But what's both?
Oh, the product at the end is, you know, each one of these, so you have the 5 carbon regeneration, and that one carbon.
I think that would be for
So you need to
for the
six
sunny and the
times
shade,
produce one.
especially. The limit water loss.
But that's, you know,
Yeah,
that's different from this.
Okay.
Yeah, so all of that is each, think about each C from CO2 goes from one carbohydrate.
All that's on like directly.
6 CO2,
But
yes.
that's probably also something like sun. So like if you super flat and all of it.
So you just the shape. So the shaping of it as well.
Yeah. Big shit. Yeah, usually that's this one I was thinking order.
Because some of them do turn slowly. Yep.
Because like in the issue for sunbar.
I mean, that's what this shape is.
Like an adjustment for like some plants. One was more tissue density, because like plants in a super warm environment, or like maybe subtropical where they're exposed to a lot of sun, can have cells for like or something like maybe 7 cells, it really thick tissue, less exposed tissue.
So they have thicker tissue, but also that tissue, if you think about it, that tissue is a lot of like mesophyll tissue. You're still increasing the chloroplast and you're absorbing chloroplast. So it's gonna be more wax.
I'm saying like aloe, right?
Yeah, so you're gonna have more cells that are more of wax material, so like they aren't there so much to do photosynthesis, but more to
Support plant.
yes. Because if you increase the mesophyll and the photosynthesis and the plant cannot hide. There's the light that is hitting it, it's gonna hit it. The more chloroplasts you have, the more they absorb that light. And if there's, blue light, the plant is going to have to deal with that even more.
Yeah, and I was thinking about too, doesn't the actual shape of the leaf impact it too? Because like if it's super flat, because I remember you talking about like if it's super flat, you're getting all those colors.
Yeah. Well, that's actually leaf movement. So you can have that move through the leaf. Because you even if you're, but so the thing when you think about it is like cacti, they don't even have leaves, right? So we're talking about a whole, they have needles, so they are going to have, and you cannot see like a cactus that's all turgid and trying to hold water in the stem that is basically the whole thing. You cannot just afford to do this. But cacti have, for example, they can have the needles. Some of them can have a lot of trichomes that are going to like refract some of the light.
Isn't there some like the shade plants? When you talk about some like specific
Yeah, like when
pathway
you
in photosynthesis?
like when you have a plant, when you see that these are indoor plants, these are outdoor plants, there's a reason for that.
Yeah.
And there are some plants that can be both, but there are some plants that not. And I think the book talks about how you have like an indoor plant kind of shade in the shade, and then you take it out. Some of them will adapt, some of them and they will suffer for a while, because when you think again about a plant, if I have a leaf adapted to shade and I take it to the light, there is little that leaf can do because it's fully developed. You don't have cell migration, you don't have any of that. So your option is kill it and grow something else that's better suited for that. You can do certain things to that leaf, but for the most part, there's not much. Okay, so let's talk a little bit, just a couple of questions about how plants and flowers. What happens when a plant? Let's talk. Okay, so what we talked about. What happens in terms of like molecules, not so much plant architecture just yet. What do you remember that happens to plant when it's exposed to too much light? Yes.
It has a thicker laminate and a lower concentration.
Yes, but let's not talk about the plant itself just yet. So how the plant changes, we're gonna get there. But what happens in a cell of a plant that is receiving
Loses a lot of water.
Remember that? Okay. Yeah.
Is there more Rubisco available?
Sorry?
Is there more Rubisco available?
In a plant that has too much light, why would that help a plant? A cell that has too much light have a lot of Rubisco.
It needs more of this, is it overexposed too much light? Or is producing too much?
I don't know. I'm asking you. You said a lot of Rubisco, so I just want to see the thought process. Why do you think you will have more Rubisco?
I think it's producing more of it because there's too much light.
So is too much light? Maybe I didn't answer. Is too much light good for a plant?
Not too much.
Yes. So what happens when it's exposed? Let's say too much. The plant is not happy. What happens? Why is the plant not happy if it has too much light? To the cell. What happens to that cell that is receiving too much light?
I think it can only do so much work, right? So if it's exposed to too much, the cellular processes are happening too often or too much.
And as a result, what happens to the cell? Do you know why? So you're hitting a key point. The plant is doing a couple of things too much, and it's producing. It's not happy. There's too much light, right? I don't know how to say. I'm talking too much light. So there are a lot of photons, high energy photons hitting that cell. What happens to that cell? How is that cell being affected?
It produces free radical oxygen that can be damaging to the plant's photosynthesis.
Yes, and so what are some of the things that plant can do to eliminate some of that, or to deal with some of that? Just mention one thing.
Carotenoids that absorb extra energy.
Yes. So basically, what is going to happen is that plants exposed to too much light, they don't like it, right? They start developing free radicals, and so they have options dissipating it, and one of the main reasons or the main options they have is using particles to dissipate. So let's do one more question. So again, in terms of think you're a plant and you're not getting enough light. How can I see a plant and say, hey, this plant is not getting enough light? How is that going to express the plant? And you're in your house and you think the plant looks like it needs more light. How can you tell maybe your plant needs more light? Yes. Yes.
Like it might become like shriveled or like kind of what's the word? Like over, like not. Yeah, misshaped. Thank you. It might be like misshaped or like slumped over, or or like the color might be off as well.
Yes. What's key about the color? Like how would you see the color change?
Is it due to the chlorophyll?
So is it gonna get like, how would you describe the difference?
I believe it would be like a lighter color.
Yes. So basically, when a plant is not getting enough light, photosynthesis is inhibited, right? You can, if you need enough carbon as a fertilizer, but remember it's all about the carbon and nitrogen ratio, so it is going to develop chlorosis, which is the pale color of the leaves. It's gonna grow slower, and it's gonna have basically just thinner leaves, because it wants to maximize light absorption, okay? Okay, so let's leave it there. Just gonna give one more try, and this is quick, I promise. If anybody wants to get a point quickly. Oh my God, I don't know. Exactly. Yeah. Yes.
Wow, thank you. Too little light, because it is a lighter color, like he just said,
Yes.
and the leaves are more spread out, and the best way to get more light.
Yes. So what did you say about the leaves spread out?
They're trying to like reach more light, trying more surface
Yes.
area.
So you're gonna see too little light, they are pale, and they are trying to get to a place where there's more light. Okay? Good. So I will see you on Friday.